In 1976, a Viking lander mixed Martian soil with nutrient solution, and the experiment gave a signal. The question is whether NASA had already run the only real life detection test on Mars, or whether Mars chemistry fooled the instrument. That is why Viking refuses to disappear. It was not a vague search for habitability. It was a direct biological experiment on Martian soil, and one part of it looked positive, immediately and uncomfortably so. But a positive-looking signal is not life. The other Viking instruments did not find the expected organics. Later missions revealed oxidants, perchlorates, and reactive chemistry in Martian soil. The result sits between biology and chemistry. Over the next 2 hours, we stay inside that contradiction until the boundary becomes clear. By the end of this video, you will understand why Viking remains unresolved in public memory, and why science has not treated it as confirmed life. If you enjoy exploring questions like this with us, subscribing helps others find them too. Sharing the video helps even more. Let's begin. Now we should treat Viking as a working lab before we treat it as a symbol. We need the machinery in view, because the machinery defines the question. The other two biology experiments show why Viking was not built around one narrow hope. Gas exchange looked for changes in the gases above a treated sample. Pyrolytic release asked whether carbon from a Martian-like atmosphere could be incorporated into organic material under light. These tests were different ways of approaching the same unknown. Maybe Martian life would respond to nutrients. Maybe it would alter the atmosphere around it. Maybe it would build carbon compounds under surface conditions. None of those assumptions was guaranteed, but together they show the mission's seriousness. Viking did not carry a decorative life experiment for publicity. It carried a suite of tests because the biological question was central to the mission's identity. Now picture what had to happen before any result could exist. The lander needed to collect soil with a mechanical arm, deliver small amounts to different instruments, keep the chambers sealed, manage temperatures, and send data across interplanetary distance. This was not a scientist holding a sample under a bench microscope. It was a robot carrying out a prepared experiment millions of kilometers from its builders. That matters because every design choice became part of the answer. The scoop could reach only nearby surface material. The chambers could create only certain conditions. The nutrients, gases, lights, and temperatures had been chosen on Earth. Viking was asking Mars a direct question, but it had to ask in a language humans had invented before meeting the sample. So the mission stands between courage and limitation. It was courageous because it did not wait for perfect context before testing biology. It was limited because the context was exactly what Mars later proved we needed. That tension begins before the first signal appears. Viking is easy to picture as a famous Mars landing. A spacecraft survives the atmosphere. A lander settles onto the surface. The first images come back from a world humans had only seen from orbit. But if we stop there, we miss what made Viking so unusual. Viking was not only a camera on legs. It was a small laboratory placed on the ground of Mars. Its job was not simply to say what the ground looked like. It was built to touch the soil, move it into instruments, change its conditions, and watch what happened next. Now think about the timing. Viking 1 landed in Cres Planitia in July 1976. Viking 2 landed in Utopia Planitia that September. These were not rover stops, they were fixed laboratories, separated by thousands of kilometers, each with a scoop, an instrument deck, and a biology package that had been designed before anyone had ever operated a lab on another planet. The biology package mattered because the question of Martian life was still open in a very direct way. Scientists did not yet have rover tracks across ancient lake beds. They did not have drilled mudstone samples. They did not have decades of mineral maps, methane debates, or perchlorate chemistry. Mars was known to be cold, dry, and harsh, but it was also a planet with seasons, polar caps, changing surface markings in older telescope lore, and a long public history of speculation. So Viking carried experiments that were willing to ask the blunt question: if a bit of Martian soil were given the right conditions, would it do something that looked biological? That directness is what separates Viking from most later Mars missions. Later missions often asked whether Mars had once been habitable. They searched for signs of water, carbon chemistry, minerals formed in ancient wet environments, preserved organics, and places where life could have existed. Those are serious life-related questions. They changed our picture of Mars, but they are not the same as placing fresh Martian soil into a chamber, feeding it, changing its atmosphere, or exposing it to carbon dioxide, then watching for a biological-style response. Viking did that. Its biology package included 3 major experiments. The labeled release experiment added nutrients tagged with radioactive carbon and watched for radioactive gas. The gas exchange experiment added moisture and nutrients, then looked for changes in gases above the soil. The pyrolytic release experiment exposed soil to radioactive carbon dioxide and carbon monoxide under simulated Martian conditions, then checked whether carbon had been incorporated into organic matter. And beside those biology tests was the gas chromatograph mass spectrometer, usually shortened to GCMS. That instrument was not another metabolism test. It was a chemistry instrument. It heated samples and searched for organic molecules, the carbon-based compounds that known life uses and leaves behind. But the GCMS still mattered enormously. A life test signal without organic chemistry support would be hard to defend. A biology experiment could show activity. The chemistry instrument could ask whether the molecular raw material of life was actually present. Here is the first important tension. Viking did not fly one magic life detector. It flew a group of instruments that could disagree. That was good science. A single response is never enough when the claim is this large. If we are going to say another planet has living chemistry, the answer has to show up in more than one way. It should appear in activity, in molecular evidence, in environmental context, and in controls that rule out ordinary chemistry. But Viking was also trying to do something almost impossible for its time. It had to design experiments for an unknown surface. It had to decide what Martian life, if it existed, might eat. It had to guess how dry soil would behave when warmed, wetted, fed, or heated. It had to distinguish biology from chemistry on a planet where the surface had been exposed to ultraviolet light, cosmic radiation, dryness, and oxidizing reactions for enormous stretches of time. Now picture the lander again. The scoop reaches down. It does not know whether it is holding lifeless dust, chemically charged dust, dormant cells, damaged organics, or some mixture nobody had imagined. The soil enters a chamber smaller than a kitchen cup. Human beings on Earth wait for numbers, and that is why Viking was brave, not because it promised a simple answer, but because it put the question into hardware. And that hardware had limits. Each experiment assumed something about life. The labeled release test assumed that possible microbes might consume a nutrient solution and release labeled gas. The gas exchange test assumed that adding moisture and nutrients might cause biological gas changes. The pyrolytic release test assumed possible organisms might take up carbon from a Martian-like atmosphere under light. These were reasonable designs. They were also guesses. We should sit with that for a moment. A life detection experiment is never just an instrument. It becomes a theory about what life will do when touched. If the theory matches the organism, the experiment can be precise. If the planet's chemistry mimics the same behavior, the experiment can be fooled. If life exists but does not respond to the food, water, temperature, or chemistry offered, the test can miss it. And if the sample is from the wrong place, the result can be true for that handful of soil and still not answer the planet. Viking stood at the beginning of that problem, but even with those limits, it remains special. No later Mars lander has operated the same kind of broad, direct biology package. Later missions became more careful, more contextual, more geologic, more molecular, and in some ways more powerful. Yet they also stepped back from the blunt metabolism question Viking asked on the surface. So when people ask whether NASA already ran the only real-life test on Mars, the careful answer begins here. If real-life test means a direct biology package trying to elicit lifelike activity from Martian material, Viking is still the mission at the center of the story. That does not make its answer simple. It makes its ambiguity more important, because among Viking's 3 biology experiments, one response became the signal that refused to disappear. There is one final practical detail. Viking's landers could not move to chase a better sample. Once each spacecraft touched down, the nearby surface became the whole reachable world. If a scoop hit oxidized dust, that was the sample. If better preserved material lay centimeters deeper or meters away, the biology package could not go looking for it. Later rovers changed that expectation by turning Mars into a crossed terrain. But Viking had to make history from what sat beside its feet. That makes its directness even more remarkable and more limited. It asked the boldest possible question with a tiny local handful of Mars. Here is also a useful distinction between direct and complete. Labeled release was direct because it tested fresh Martian material for activity. It was not complete because no single activity test can capture every requirement for life detection. A direct clue can still need indirect support. In fact, the more direct the clue feels, the more carefully it needs to be checked, and that is why the experiment remains so powerful in memory. It was direct enough to feel like Mars answering. It was incomplete enough to keep scientists arguing about the voice. The radioactive tag can sound dramatic, but for the experiment it was just a way of following carbon. Without the tag, a little gas released in a tiny chamber could be hard to connect to the food. With the tag, the instrument could ask whether the carbon coming out was the same carbon that had gone in. It made the result cleaner at the detector level, even if the interpretation stayed difficult. Now think about why a gas response is such an attractive clue. Gas moves. It can leave the soil and enter the detector. You do not need to dig through grains looking for microscopic structures. You can let the sample transform a molecule, then watch the product arrive. For a remote spacecraft, that is powerful. But it also means the experiment mostly sees an ending, not the whole process. It sees labeled gas after nutrient addition. It does not see every molecular step between. That hidden middle is where the dispute lives. One reason the design still grabs attention is that it was wonderfully simple at the listening level. The laboratory did not have to recognize a Martian cell. It did not have to know what Martian genetics might look like. It did not have to guess whether life on Mars would use the same molecules as life on Earth. It asked for behavior. Give the sample a small chemical opportunity, then monitor whether carbon from that opportunity returns as gas. Now, that simplicity was also the danger. A test based on behavior has to know what else can behave. On Earth, we often separate life from mineral chemistry because the background is familiar. We know what ordinary soil does under many conditions. We know how sterilized soil behaves. We know how microbial communities respond to nutrients. Mars did not come with that library of experience, but the labeled release team had a strong reason for choosing this approach. Life, whatever its deeper biochemistry, must exchange matter and energy with its environment. It has to do something. It has to transform something. A fossil can be silent. A dormant cell can be quiet. But metabolism, if present and stimulated, should leave a trace. The experiment tried to make that trace detectable with the smallest possible ambiguity in the detector itself. Tagged carbon in, tagged carbon out. So when we call it metabolism-style, we are not saying the instrument saw metabolism directly. We are saying the test was designed around the kind of exchange metabolism would produce. It was a behavioral assay. It asked whether the soil could turn offered carbon into a released gas under the conditions Viking supplied. And those conditions matter. The nutrient solution was not a Martian meal designed by Martians. It was an Earth-designed mixture of simple organic compounds. If Mars life existed but used a very different chemistry, or if it was damaged by the added water, or if it lived below the surface and not in the oxidized dust, the test might miss it. A negative result would not have ruled out all life everywhere on Mars. And that is another reason the actual positive-looking result became so interesting. It did not answer every life question. It answered the question the instrument asked: does this sample produce labeled gas after receiving labeled nutrients? The answer was yes, enough to force interpretation. Now think about the detector as a kind of translator. The soil speaks in chemistry. The instrument translates only one narrow phrase: labeled gas leaving the sample. If the phrase appears, we still have to ask who spoke it. Was it a living process? Was it reactive mineral chemistry? Was it a combination of wetting, oxidation, and nutrient breakdown? The translation is clear, but the speaker is uncertain. That image helps keep us honest. The labeled release result is not nothing. A translator heard a phrase, but the phrase did not name its author. Now let's slow the labeled release experiment down until it becomes physical. Imagine a tiny cup of Martian soil inside the lander. Into that cup goes a small amount of liquid nutrient. The nutrient contains simple organic food molecules. The carbon in those molecules carries a radioactive label, not because the experiment is trying to harm anything, but because the label makes the carbon easy to track. If something in the soil consumes the nutrient and releases carbon as gas, the instrument can notice. The gas would carry the radioactive tag. The detector would not see a microbe. It would not photograph a cell. It would not hear a heartbeat. It would measure labeled carbon leaving the sample, and that is the beauty of the design. It turns a hidden biological possibility into a number. On Earth, a result like that can make sense. Soil is full of microbes. Feed them carbon compounds, and some will metabolize the food. Gas comes out. The exact details depend on the organisms and the chemistry, but the basic idea is familiar. Living systems take in matter, transform it, and release waste. So the labeled release experiment was listening for a breath. Not a human breath, not an animal breath, a chemical breath. A release of labeled gas after a Martian soil sample received tagged food. But we have to be precise. A breath-like response is not automatically life. Chemistry can also transform molecules. Reactive minerals can break compounds apart. Oxidants can attack nutrients. Wetting dry soil can trigger reactions. Heating a control sample can change more than biology. On an alien surface, the same detector can be reading a living process or a chemical imitation. And that is why controls were central. In the basic logic of the test, an active sample gets the nutrient and produces a response, if something in the soil can make that happen. A control sample is treated in a way expected to remove biological activity such as heating. If the heated sample no longer responds in the same way, that difference can support a biological interpretation. But the control is not magic. Heat can kill microbes. Heat can also change minerals, destroy oxidants, release adsorbed gases, alter reactive salts, or rearrange the chemistry of the soil. So when a heated control behaves differently, the difference matters, but it still has to be interpreted, and here is the hard part. Viking was doing this on soil no human had ever held. We know terrestrial soil by experience. We know it can be alive. We know it carries organics. We know what many controls mean in Earth laboratories. Mars was different. Its surface was dry, cold, irradiated, and exposed to intense ultraviolet light through a thin atmosphere. Its chemistry had been processed under conditions that do not resemble a garden, a forest floor, or a wet field. So the labeled release experiment carried Earth logic to Mars, then waited to see whether Mars would play by the same rules. But the experiment was not naive. It had been tested with Earth soils, including harsh environments. The team knew that metabolism is not the only possible gas source. They built in controls, repeat runs, and interpretation criteria. The result would not be judged only by whether any gas appeared. Timing mattered, the amount of gas mattered, the effect of heating mattered, repetition mattered, comparison between landers mattered. Now picture the first active run. The sample is inside the chamber. The nutrient touches the soil. If the line stays flat, the story becomes one kind of disappointment. If the line rises, everything changes. The line rose. That simple statement carries the whole debate. A gas release after nutrient addition was exactly the kind of response the experiment was built to watch for. The signal appeared quickly enough and strongly enough to matter. It was not a vague instrument twitch that could be ignored before coffee. It was the sort of result that made the scientists responsible for the experiment take the biological readings seriously. And because the carbon was labeled, the instrument had a clean target. It was not asking whether some random gas shifted in the chamber. It was watching for the tagged carbon from the nutrient to leave as gas. In plain language, the food went in and labeled gas came out. And that is why this test still feels different from later Mars life discussions. A rover finding ancient organic molecules is important, but it is not the same thing. Methane changing in the atmosphere is intriguing, but it is not the same thing. Minerals formed in water tell us about past habitability, but they do not show a present sample responding to food. Viking's labeled release test was direct in a way that still sounds startling. But direct does not mean decisive. If we were dealing with Earth soil, a positive response plus a strong sterilized control might quickly lean toward biology. On Mars, the same pattern had to compete with a stranger possibility, a planet whose soil chemistry had stored enough reactivity to behave like something alive when the nutrient arrived. And that is the difference between a signal and a conclusion. The signal is what the instrument measured. The conclusion is what scientists decide the signal means. In this story, those two things never fully merged. Still, before we look at the objections, we need to give the signal its strongest fair hearing, because the debate survived for decades not because people ignored the data, but because the data had structure. The instrument did not stay quiet. It answered. Now we have to give the signal its fair weight. We can be skeptical without pretending the curve was empty. We can be careful without draining the result of its force. If we listen carefully, the strongest pro-life argument is not believe the exciting version. It is do not erase the instrument that gave the exciting result. That is a fair demand. The data deserve to be included, not explained away by tone or embarrassment. The strongest skeptical answer is not nothing happened. It is something happened, but life is not the only cause. That is also fair. The difference between those two sentences is the whole documentary. Here is one more reason the signal felt so difficult to put away. Viking did not arrive expecting Mars to be generous. The environment already looked harsh. A positive-looking biology result in that setting was not an easy, comfortable claim. It cut against the dead surface picture, which made it both exciting and suspect. For proponents, that made the result more important. If a test built for metabolism responds on a hostile planet, maybe the hostility is not the whole story. Maybe life is tougher, stranger, or more localized than the surface view suggests. Maybe the experiment had caught a fragile biological trace before other instruments could understand it. For skeptics, the same hostility made chemistry more likely. A surface that harsh is exactly where unusual oxidants and radiation-driven reactions might dominate. The very conditions that made biology surprising also made false positives plausible. That symmetry is why the debate never becomes easy. The timing of the Viking mission made this even more charged. In the 1970s, there was no long rover record to place the result inside. No detailed mineral maps from orbit, no drilled cores from ancient lake deposits, no Phoenix perchlorate discovery. The labeled release curve arrived in a young science, and it arrived with the emotional force of first contact with Martian soil. But emotional force is not the same as scientific weakness. The reason the curve mattered is that it answered the experimental design. A metabolism-style experiment expects a change after food is added. The change occurred. That is exactly why the debate is serious. Now, proponents often stress that the response was not one mysterious burst. They argue that its shape, its timing, and its reaction to later treatments fit better with biology than with the chemical explanations proposed at the time. They also argue that some Earth soils with microbial life produced comparable results, while sterilized versions did not. In their view, Viking saw the thing it had been sent to see. The mainstream answer is not that these details are imaginary. The answer is that similarity is not identity. A Martian reaction can resemble a biological pattern without being caused by organisms. The shape of a curve can be suggestive and still not unique. Heat sensitivity can suggest life and still be shared by non-living reactive compounds. Repetition across two sites can show a widespread Martian soil property rather than widespread biology. So the fairest reading has to hold two uncomfortable ideas together. The positive-looking result had enough structure that serious scientists could defend a biological interpretation. The same structure did not exclude the chemistry of a dry, irradiated, oxidized planet. Here is the kind of ambiguity that science hates and needs. It hates it because ambiguity blocks the announcement everyone wants. It needs it because ambiguity reveals where the next instrument must improve. Viking did not only produce a contested data set, it exposed the exact weaknesses in how a direct life test can be interpreted. Now imagine the public version of this moment: a lander on Mars, a direct biology experiment, a signal after nutrients, then caution. To many people, caution can feel like retreat. It sounds as if the exciting result was being softened. But inside the science, caution was not an emotional choice. It was a response to the size of the claim: if there is life on Mars, the discovery changes biology, planetary science, and our sense of life in the universe. That kind of claim has to survive more than one instrument's interpretation. It has to survive the strongest non-living explanation people can build. Viking's signal did not survive that test cleanly, but it also was never explained away so completely that everyone stopped talking. And that is why this result has such a strange afterlife. It's too provocative to bury. It's too incomplete to crown. At both Viking landing sites, the labeled release experiment produced positive-looking responses. That matters. Viking 1 and Viking 2 were not sitting side by side. They landed in different regions of Mars, separated by a planetary distance. Viking 1 touched down in Chryse Planitia. Viking 2 landed in Utopia Planitia. Each site had its own local surface, its own soil sample, its own environmental setting. So when the labeled release experiment gave a response at both sites, the result became harder to dismiss as a one-off glitch. It did not prove biology, but it gave proponents something serious to work with. Now imagine being on the science team. You built a test to look for metabolism-style activity. The test adds labeled nutrients. Gas appears. A control behaves differently. Then another lander, far away, sees a related pattern. You would not shrug that off. You would ask whether the experiment had done what it was built to do. Gilbert Levine and Patricia Straat, closely associated with the labeled release experiment, argued for years that the result met the experiment's pre-flight criteria for life detection. Their case leaned on several features: the rapid gas release, the repeated positive-looking responses, the behavior of heated controls, and comparisons with terrestrial soils. We do not have to accept that conclusion to respect why it was made. The first persuasive feature was speed. The response came soon after the nutrient was added. In a biological reading, that looks like active chemistry ready to use the food. Not a fossil, not a slow geological process over centuries. A sample in a chamber responding on the timescale of an experiment. The second feature was repetition. Both landers produced the kind of response that kept the argument alive. Repetition is one of the basic ways science protects itself from accidents. One instrument run can mislead. Two distant sites with related behavior deserve more careful thought. The third feature was the control behavior. Heated samples did not behave like the active samples. Proponents saw that as consistent with biology, because heating should damage or kill living organisms. A living system should be sensitive to sterilizing heat. A purely chemical system might not show the same pattern, depending on the chemistry. But that last phrase is where the uncertainty enters, depending on the chemistry. Mars soil was not a known laboratory reagent. It was not an Earth garden sample with familiar microbial communities. It was material from a dry, oxidized, ultraviolet processed surface. Heating could alter biological activity, yes. It could also change the non-biological reactivity that made the soil produce gas in the first place. So the heat control mattered, but it did not settle the case. Still, the pro-life argument had another emotional force. The labeled release experiment was one of the only Viking biology tests whose result looked, at first glance, like the answer people had hoped for. It behaved in the direction a direct metabolism test was meant to notice. If all Viking instruments had produced flat nothing, the story would have faded. If all instruments had agreed on life, the story would have exploded. Instead, one instrument gave a result that looked alive enough to argue over. And the people who argued for biology were not simply making a public relations claim. They were pointing to flight data, control patterns, test criteria, and Earth analogues. They believed the burden had been unfairly shifted after the GCMS failed to find organics. In their view, the biology instrument had spoken, and the negative chemistry result did not deserve to silence it completely. Now we need to separate two things. One is the strongest fair version of the biological case. The labeled release experiment gave positive-looking responses at both sites. The responses appeared after nutrient addition. Controls changed the behavior. Terrestrial tests had shown that similar methods could detect life in Earth soils. Later discoveries made Mars less chemically barren than many people assumed in the late 1970s. The other is the scientific standard for proof. A life claim on another planet cannot rest only on one line of activity, especially when the sample environment is chemically unfamiliar. It needs independent confirmation. It needs molecular support. It needs controls that isolate biology from reactive chemistry. It needs a broader context that makes the interpretation hard to counterfeit. The Viking result had the first part. It did not have enough of the second, but let's not rush away from why the result felt so powerful. The response was not a philosophical possibility. It was a number coming from an instrument on Mars. A machine had touched the soil with labeled food and recorded gas. So that is why people still talk about it. If a modern mission produced the same result tomorrow, headlines would explode before the second instrument even warmed up. The difference is that modern scientists would immediately demand more. They would ask for organics. They would ask for molecular patterns. They would ask for controls against oxidants and perchlorates. They would ask where the sample came from and whether contamination had been excluded. They would not let one response carry the word life by itself. Viking taught us that lesson the hard way. The biological case survived because the signal was real enough to respect. The mainstream hesitation survived because the surrounding evidence made the interpretation unsafe, and the most important surrounding evidence came from the instrument asking a colder question. Where was the carbon? So we keep both instruments in the room. We need the LR signal, and we need the missing organic support. Now we have to let the chemistry instrument speak just as clearly as the biology instrument. We cannot choose only the measurement we like. We need the disagreement, because the disagreement is the story. Here's also why the Viking debate became a model for biosignatures more broadly. A biosignature is a possible sign of life. The word possible is doing a lot of work. Oxygen in an exoplanet atmosphere can be biological or non-biological. Methane can be biological or geological. Organic molecules can be made by life or delivered from space. A pattern becomes stronger when non-life routes fail. Viking's LR result was an early biosignature problem before the term became common in public discussion. It showed that the sign and the source are not the same. The word organic also needs care. In everyday language, organic can sound biological or natural. In chemistry, it usually means carbon-based compounds. A molecule can be organic and have nothing to do with life. Meteorites carry organics. Geological reactions can make organics. Laboratory contamination can introduce organics. Life uses organics, but organics alone do not equal life. So the GCMS was not looking for a single life molecule. It was looking for the carbon chemistry that would make a life interpretation more believable. If it had found a rich and convincing organic inventory, scientists would still have needed caution, but the labeled release result would have stood in a very different setting. Instead, the chemical picture looked empty by the expectations of the time. That emptiness became the great counterweight to the gas signal. The organics issue also changed the burden of explanation. If you argue for biology in the labeled release chamber, you have to explain why the chemistry instrument did not see the expected carbon inventory. If you argue for non-biological chemistry, you have to explain why the activity signal looked so lively. Neither side gets a free answer. Now, known life is more than carbon sitting in a sample. It organizes carbon in patterned ways. Cells do more than contain random organic molecules. They build structures, maintain gradients, and copy information, harvest energy, and leave chemical traces. A living soil should not be chemically indistinguishable from mineral dust in every other instrument. So the lack of expected organics was a real strike against the biological reading. But the Viking surface was not a gentle archive. Organic molecules delivered by meteorites, made by geological reactions, or left by hypothetical organisms would have to survive radiation, oxidants, dryness, and then the analytical process inside the instrument. If they were destroyed at the surface or transformed during heating, a non-detection could be telling us about preservation rather than absence. And that is why later discoveries became so important. They did not rescue the life claim. They changed the meaning of the old chemistry objection. A result that once sounded clean began to sound conditional. No expected organics were detected by that instrument in those samples under those heating conditions. Within its limits, interpreted with what was known then, that longer sentence is less tidy, but it is more accurate, and Mars often punishes tidy sentences. The GCMS also teaches a broader lesson about supporting evidence. A biology experiment can be designed around activity, but life is not only activity. Life is material. It has molecules. It changes its environment. It leaves patterns. The stronger the claim, the more those different signs should support one another. Viking did not get that support, and for us that means the disagreement is not a side note. It becomes the central object we have to hold in view if we want to understand why the answer stayed open. So when NASA and many scientists held back from a life announcement, they were not ignoring the labeled release result. They were weighing it against a missing partner. In a weaker claim, one strong instrument might be enough. In a life claim on another planet, disagreement between instruments becomes decisive. Now, that decision did not make the labeled release team vanish. It sharpened their objection. They could say the chemistry instrument had its own assumptions and limits. They could say the biology experiment should not be demoted simply because another instrument failed under alien conditions. They could ask whether the negative result was being treated with more confidence than it deserved. That tension is healthy. It's how science works when no single line of evidence controls the whole story. The mistake is to turn that tension into heroes and villains. The labeled release team was not foolish for taking the results seriously. The broader community was not cowardly for demanding organics and independent confirmation. The instruments disagreed because Mars gave them a hard problem. And once the soil itself became suspect, the problem became even harder. If the labeled release experiment was Viking's dramatic voice, the GCMS was its skeptical nose. The gas chromatograph mass spectrometer was built to search for organic molecules in Martian soil. In ordinary language, it was looking for carbon-based compounds. Not life itself, not cells, not metabolism, but the kinds of molecules known life is made from, uses, breaks apart, leaves behind, and reshuffles constantly. On Earth, life is carbon chemistry. Every living cell is crowded with organic molecules. Even dead organisms leave chemical remains. Soil with active life is not chemically empty. It carries carbon compounds from cells, waste, membranes, proteins, sugars, fats, pigments, and decay products. So when Viking's GCMS did not find the expected organic inventory in the surface samples, the life interpretation ran into a wall. Now, this is where the story is often flattened too much. The organics problem was not a bureaucratic excuse. It was not a committee looking for a way to avoid an uncomfortable conclusion. It was a serious scientific objection. If one experiment suggests metabolism, but another instrument cannot find the carbon chemistry that known metabolism requires, scientists have to hesitate. They should hesitate. A positive-looking activity signal without the expected molecules is exactly the kind of contradiction that separates evidence from proof. But the GCMS result also needs careful wording. The instrument was not an all-powerful detector. It had limits. It used heating. It operated with 1970s technology. It sampled the oxidized surface. It was looking under assumptions about what organics should survive, how they would behave during analysis, and how to distinguish Martian compounds from contamination. Later work complicated the story even more by showing that heating perchlorate-bearing samples can change or destroy organics and produce chlorinated compounds. So the GCMS did not become the final word on Mars life either, but in 1976 it was devastating to the life claim. Think about the evidentiary balance. One instrument says the soil released labeled gas after receiving nutrients. Another instrument does not find the expected organic molecules. The first result says something in the soil was chemically active. The second says the material did not look like living carbon chemistry in the way scientists needed. That disagreement is not a small footnote. It becomes the core of the Viking mystery. If the GCMS had found abundant organics, the labeled release result would have looked very different. It still would have needed controls. It still would have needed caution. But the case for biology would have had a molecular partner. Instead, the biology-style signal stood almost alone. Now picture the situation as a courtroom, but without villains. The labeled release result walks in with a strong claim. I saw activity after food. The GCMS walks in with a hard objection. I do not see the carbon inventory that should support that claim. Neither instrument is lying, neither is covering for the other. They are measuring different things. The difficulty is that Mars gave them results that did not compose a clean sentence. For NASA and much of the planetary science community, the organics non-detection made it unsafe to declare life. That caution was not anti-science. It was science doing what it is supposed to do when a result is extraordinary and incomplete. But for labeled release proponents, the GCMS objection also had weaknesses. They argued that the biology experiment should not be dismissed simply because a chemistry instrument failed to find organics under harsh analytical conditions. They argued that detection limits, sample treatment, and unknown Martian chemistry could matter. Later, perchlorate research gave that objection more weight than it had at the time. So the organics problem did two things at once. It blocked proof, and it did not erase the signal. That double role is easy to miss. People want the GCMS to be either the debunker or the failed instrument. It was neither. It was a necessary cross-check that made the life interpretation much harder, while also becoming part of a larger question about what Martian surface chemistry does to organics when heated. And there was another clue. The surface of Mars was not chemically gentle. Even before Phoenix discovered perchlorate decades later, scientists were thinking about reactive oxidants. Viking results suggested that Martian soil could contain substances able to destroy organic molecules and produce unusual reactions. The planet's surface was bombarded by ultraviolet light. It was dry. It was exposed. It had no thick atmosphere or global magnetic shield like Earth's surface environment. Chemistry there could become aggressive in ways that make simple Earth analogies dangerous. Now we can see the problem more clearly. The labeled release experiment may have detected something active. The GCMS did not find the expected organic support. A chemically reactive surface could help explain both. It could attack the nutrient and release gas. It could destroy or alter organics. It could change when heated, creating control patterns that resemble biology but are not biology. That possibility became the mainstream non-biological lane, not because scientists wanted Viking to be boring, because the evidence file pointed toward a planet whose soil was not passive. The next step is to understand that soil. Dead does not always mean inert. The soil chemistry explanation also has to be kept honest. It should not become a magic trash bin where any inconvenient result disappears. A good abiotic model needs details. What compound reacts? What gas is produced? Why does heating change the response? Why do both sites behave similarly? How does the model fit the GCMS evidence? That demand keeps the debate scientific. Biology has a burden. Chemistry has a burden too. This also changes how we imagine the Viking sample. It was not a neutral witness carried into court. It was an active participant. The moment the nutrient arrived, the sample's minerals, salts, adsorbed gases, and reactive surfaces all had a chance to respond. The test did not simply reveal Mars. It created a new Martian microenvironment inside the lander. That microenvironment may have been unlike anything naturally present at the landing site. Liquid nutrient touched dry, oxidized dust. A chamber held gases, temperatures were controlled. The sample was isolated from wind, sunlight, and the broader surface. If life was there, maybe that helped it. If chemistry was waiting, maybe that helped chemistry too. The instrument created the stage. The soil supplied the actor. We still disagree about what kind of actor it was. One helpful way to picture this is to forget Mars for a moment and think about household chemistry. Pour vinegar onto baking soda and gas appears. Add hydrogen peroxide, the right catalyst, and bubbles rise. Wet certain dry materials and heat comes out. None of that requires life. It requires stored chemical potential. Mars may store chemical potential in its surface in unfamiliar ways, not because it is magical, but because its environment is different. A thin atmosphere, intense ultraviolet exposure, low temperatures, dryness, and long time can create and preserve reactive materials near the surface. The soil can be quiet until the experiment changes its conditions. Viking changed the conditions. It scooped, it sealed, it warmed, it added liquid, it added organic nutrients, it heated controls. Each action was meant to reveal life if life was present. Each action could also disturb a non-living chemical system, and that is the deep design problem. An experiment is not a window that looks without touching. It touches, it changes the sample, it creates an environment that did not exist before. The result is a conversation between the original soil and the conditions the instrument imposes. For Earth soil, that conversation is easier to understand because scientists have heard it many times. For Mars, Viking was one of the first listeners. Now, if reactive soil chemistry explains the labeled release result, Viking still made a discovery. He discovered that Martian surface material could respond strongly enough to imitate one of life's expected behaviors. That is not as romantic as microbes, but it is scientifically enormous. It tells future missions that the planet can counterfeit a sign we care about, and counterfeit is the right idea only if we use it carefully. The soil is not trying to fool us, it has no intention, it simply follows chemistry. We are the ones who built an instrument where two causes could look similar. Here is why the phrase dead soil can mislead. Dead suggests inactive, but non-living matter can be reactive, patterned, energetic, and surprising. A lightning storm is not alive. A hydrothermal vent is not alive. A chemical battery is not alive. Mars dust does not need cells to do chemistry. So the real opposition is not alive versus dead. It's biological metabolism versus abiotic reactivity. That vocabulary sounds more careful because it is more careful. It keeps us from imagining Mars as either a garden or a corpse. The surface can be hostile to life and active in chemistry at the same time. Now return to the heated controls one more time. If heat changed a reactive oxidant, then the control result can mimic sterilization. If heat destroyed a fragile biological system, the same control result can support life. The observation is shared, the cause is contested. So that is why the Viking debate becomes so stubborn. The correct response is not to pick the simpler story too early. It's to ask what additional chemistry would make one cause more likely than the other. Decades later, perchlorate gave scientists one of those additional clues. When most people hear soil, they imagine something Earth-like. Dirt, dust, sand, maybe clay. Something that sits there until a seed, a root, a worm, a raindrop, or a shovel disturbs it. Mars soil is not like that. The upper surface of Mars has spent immense time under conditions that are rough on chemistry. The air is thin, liquid water is unstable at the surface today. Ultraviolet radiation reaches the ground more strongly than it does on Earth. The planet is cold and dry. Dust can move across the surface, but there is no global biosphere constantly refreshing the soil with plants, microbes, moisture, and organic decay. That environment can make the regolith chemically reactive. Regolith is the loose material covering rock, dust, grains, broken minerals, and surface particles. On Mars, that material is not passive powder. It has been processed by radiation, dryness, oxidizing conditions, and interactions with the atmosphere. Scientists have long considered the possibility that reactive oxidants in the surface material could destroy organics and create chemistry that imitates biological activity. An oxidant is a chemical that can take electrons from other substances. In everyday terms, it can drive reactions that break molecules apart or transform them. Oxygen rusting iron is one familiar version of oxidation. Mars is literally the red planet because iron-bearing minerals have been oxidized. But the relevant surface chemistry can be more subtle and more reactive than rust-colored scenery. Now bring that into the Viking chamber. A dry, reactive soil sample sits there. A liquid nutrient arrives. The sample is suddenly wetted. Organic molecules are suddenly available. Temperature conditions change. Gases can be released. Reactive compounds can attack the nutrient. Minerals can adsorb, desorb, break, or transform molecules. To an instrument watching for gas that can look active. And here is the uncomfortable part for life detection. Activity is not the same as biology. A battery is active, bleach is active, rusting metal is active, fresh cement can react, hydrogen peroxide can foam. None of that is alive. The question is whether the activity carries the pattern, selectivity, context, and molecular support of living systems. Viking's labeled release result showed activity in the direction of a metabolism-style signal, but a chemically awake soil offered another way to get there. Now think about controls again. Heat is often used to separate biology from chemistry because living cells are fragile. Heat can damage proteins, membranes, enzymes, and the organized machinery of life. If a heated sample stops responding, that can support biology. But heat also changes non-living chemistry. It can decompose oxidants. It can drive gases out of minerals. It can alter salts. It can change surfaces and destroy reactive compounds. It can turn one chemical system into another before the test even begins. So, a heat-sensitive response does not automatically mean life. It means the response depends on something heat changes. That distinction is small in words and enormous in science. The Viking debate lives inside it. If the heat labile factor is living metabolism, the labeled release result is extraordinary. If the heat labile factor is a reactive chemical species in the soil, the result is still important but not biological. In both cases, the instrument measured something real. The fight is over the cause, and Mars had reasons to create chemical causes. The surface is dry enough that reactive compounds can persist in ways they might not in wetter environments. Ultraviolet light can produce oxidizing species. The thin atmosphere lets radiation and photochemistry matter at the ground. Dust grains can hold reactive surfaces. Over long times, even small processes can charge the upper soil with chemistry that is unfamiliar to an Earth-based intuition. Now imagine giving that material a tiny drink. For a living microbe, water and food might begin metabolism. For reactive oxidized dust, water and organics might begin a chemical attack. Both can release gas, both can change after heating, both can appear quickly, both can mislead if the detector only sees one outcome, and that is why the non-biological explanation is not a lazy dismissal. It becomes a serious answer to the same data. But the chemistry explanation also had to earn its place. It could not simply say Mars is reactive and declare victory. It needed mechanisms. It needed to explain why the response appeared, why controls behaved differently, and why expected organics were missing. It needed to connect the biology experiment and the chemistry instrument without pretending either one never happened. For years, scientists proposed oxidants, peroxides, superoxides, ultraviolet processed minerals, and other reactive surface chemistry. Some ideas were stronger than others. None instantly closed the case, but together they shifted the question away from a simple life or no life result and toward a more difficult design problem. What if Mars can counterfeit one of life's simplest signs? And that is the failure case for any direct metabolism test. Not a scary failure case, a scientific one. The instrument is honest, the sample is real, the response is measured, yet the planet supplies a chemical imitation that fits enough of the test to confuse the result. Now we can see why later missions became cautious. They were not running from Viking, they were learning from it. If a surface can be chemically active without being alive, then the next test needs more than one response curve. It needs molecular evidence, it needs context, it needs controls built around the actual chemistry of Mars, not only Earth assumptions about soil. And decades after Viking, Mars supplied a chemical clue that made that lesson sharper. A salt found by Phoenix reopened the old data. So we should treat perchlorate as context, not as a verdict. We need it because it makes the chemistry argument physically clearer, while we still leave the life question bounded. Now we can see why a later soil discovery matters to an older experiment. We are not changing Viking's numbers. We are changing what those numbers might mean in Martian chemistry. The most important effect may be psychological. Perchlorate teaches us that a planet can hide complexity inside what looks like simple dust. A scoop of red soil is more than a scoop of red soil. It may contain salts that change under heat, oxidants that attack organics, minerals with reactive surfaces, and chemical histories written by billions of years of exposure. After Phoenix, the Viking sample feels less like dirt and more like an unknown reagent. Perchlorate is especially useful as a teaching clue because it shows how later knowledge can change old evidence without rewriting history. Viking did not know to design its interpretation around perchlorate. Phoenix did not travel back in time and alter the data. The measurements stayed the same. The possible meanings changed. And that is common in science. An old observation can sit for years under one interpretation, then a new discovery changes the background chemistry. The data are not fake. The early scientists were not stupid. They were working without a missing piece. For Viking, perchlorate was one of those pieces. It made the old GCMS result less straightforward and the non-biological activity explanation more concrete. It deepened the mystery while also making life harder to claim. Perchlorate also matters because it links surface habitability to instrument behavior. On Earth, some microbes can use perchlorate chemistry in their metabolism, but that fact should not be imported casually to Mars. The Viking question is not whether perchlorate can ever be involved with life. It's whether perchlorate-bearing Martian soil could have changed what Viking measured. That distinction keeps the evidence in bounds. In the heated GCMS experiment, perchlorate can be a problem because the instrument warms the sample to release and identify compounds. If the sample contains reactive salts, heating does not simply reveal the original chemistry. It can create new products. It can burn or chlorinate organics. It can make the analysis look poorer or stranger than the sample was before heating. Now think about how quietly this changes the old argument. The GCMS result was once heard as a direct absence, no expected organics. After perchlorate, it can also be heard as a process question: what happened to organics during analysis? That does not guarantee organics were there, it only means the instrument and sample chemistry were interacting. The same process mindset helps with the biology experiments. When the labeled release nutrient entered the soil, it did not enter a neutral setting, it entered a chemically prepared surface. If oxidants or reactive chlorine species were available, the nutrient itself could become fuel for non-biological reactions. A gas signal could result from chemistry awakened by water and organics. So perchlorate sits between the two Viking puzzles. It can help explain why expected organics were hard to see, and it can support the idea that the soil had enough chemical power to imitate activity. But let's keep the guardrails tight. Phoenix found perchlorate in Martian Arctic soil, not inside Viking's 1976 sample cups. Scientists infer broader relevance because perchlorate and related chemistry appear to be part of Martian soil chemistry. But exact local conditions at the Viking sites remain a question. The existence of perchlorate on Mars strengthens a mechanism. It does not retroactively measure every Viking sample, and that is one reason the debate can continue honestly. A strong abiotic explanation does not need to be perfect to block proof. It only needs to be plausible enough that biology is no longer the only reasonable cause. For a life claim, that is enough to force restraint. But for a historical mystery, imperfect explanations leave room for argument. Now, modern reanalyses have made the non-biological case more developed than it was in 1976. They can cite perchlorate, hyperchlorite pathways, peroxide-like oxidants, UV-generated reactive species, and the way wetting and heating change these systems. The chemistry side is not simply saying, maybe minerals did it, it is building mechanisms. The pro-life side can answer that mechanisms must match the actual kinetics and controls, not simply exist in principle. That is a fair demand. A proposed chemistry has to reproduce the result, not only sound plausible, and because the original mission cannot be rerun with the same samples and modern instruments, some uncertainty remains locked into history. And that is part of what makes Viking so haunting. The samples are gone, the landers are silent, the data remain. Later chemistry can reinterpret them, but it cannot open the old cups again. Phoenix landed near the Martian Arctic in 2008 and found perchlorate in the soil. The word sounds technical, but the idea can be made plain. Perchlorates are chlorine and oxygen salts. They can be stable under some conditions and reactive under others. Heat them with organic material, and they can change what the instrument sees. They can help destroy organics. They can produce chlorinated compounds. They can turn a clean chemical question into a trap. For Viking, that mattered because the GCMS had heated Martian samples while looking for organics. Now think back to the old contradiction. The labeled release experiment saw a positive-looking response. The GCMS did not find the expected organic inventory. At the time, small chlorinated compounds detected by Viking, such as chloromethane and dichloromethane, were often treated as likely terrestrial contamination. The larger conclusion was that Martian surface samples did not show the organic molecules scientists expected. Phoenix changed the context. If perchlorates were present in Martian soils, then heating a sample in a GCMS could alter or destroy organics during the very process meant to detect them. It could also produce chlorinated organic compounds that look different from the original molecules. That does not mean Viking secretly found a rich organic soil. It means the old non-detection became more complicated. In 2010, Rafael Navarro Gonzalez and colleagues studied perchlorate chemistry with organic-bearing analog soils and showed that heating in the presence of perchlorate could produce chlorinated compounds similar to those that had puzzled Viking interpretations. This kind of work did not prove that Viking samples were full of organics. It showed a plausible pathway by which organics could be transformed or hidden during analysis, and that is a major turn. The GCMS objection still mattered, but it was no longer as simple as the instrument found no organics, so the surface had none. The instrument's method, the sample chemistry, and the heating process all became part of the evidence. Now bring perchlorate back to the labeled release side. Perchlorate itself is not a magic explanation for every Viking biology result. But perchlorate chemistry sits in a larger family of oxidants and reactive species that make abiotic responses more credible. If Mars soil contains reactive salts and oxidizing compounds, then adding a liquid nutrient can trigger chemistry. Heating controls can alter that chemistry. Organic molecules can be attacked or transformed. Gas may come out. In other words, perchlorate helped make Mars less like inert dust and more like a reactive chemical system. And that is not the same as making Mars lifeless. This distinction matters. A strong non-biological mechanism does not prove there is no life on Mars. It means one old positive-looking signal can be explained without requiring biology. That is a very different claim. Science often moves this way. It does not always answer the largest question. It narrows what a particular piece of evidence can support. More recent work, including modern re-examinations of Viking-style chemistry, has continued to argue that perchlorates and other abiotic oxidants can plausibly account for the biology experiment results. The careful version is not Viking was worthless. The careful version is Viking measured real chemical behavior, but Martian oxidants may be sufficient to explain it. That word may carries weight. It leaves room for dispute. It also prevents overclaim. LR proponents can still argue that the kinetics, controls, and repeatability fit biology better than the proposed chemistry. Abiotic proponents can answer that the soil was chemically unfamiliar, that later discoveries support reactive surface models, and that independent organic evidence was missing. Both sides are arguing about the same uncomfortable fact. One signal was never enough. Now we can see why perchlorate is such a quiet twist. It does not erase the labeled release response. It does not turn Viking into proof of a dead Mars. It does not settle whether microbes ever lived or live now, but it gives the non-biological interpretation a physical backbone, and it reaches across the old contradiction. Why might an organic sniffing instrument fail to show the expected molecules? Because heated chemistry may have altered or destroyed them, especially if reactive salts were present. Why might a soil sample release gas after nutrient addition? Because reactive oxidants could attack added organics. Why might heating a control matter? Because heat changes both life and chemistry. Suddenly, Viking looks less like a failed yes or no test and more like the first accidental lesson in alien analytical chemistry. That lesson shaped everything after it. But Mars itself also changed in our imagination. The post-Viking picture did not stay frozen in the old language of a barren oxidized desert. Later missions found evidence of ancient water, habitable environments, organics preserved in rocks, and unexplained methane variability. Those discoveries did not prove Viking biology, but they made the planet harder to dismiss. Mars became less barren than the simplest Viking-era story allowed. Here's the modern Mars that makes the Viking question worth a long-form treatment. Not a planet secretly proved alive. Not a planet dismissed as dead. A planet where habitability, preservation, destruction, and ambiguity all exist together. And that is much harder to explain than a headline. Also, much closer to why Mars still matters. The richer Mars also gives the viewer a better reason to care about an old lander. Viking is more than a museum story. It becomes the beginning of a question that Curiosity, Phoenix, Perseverance, and future missions still live with in different forms. How do we recognize biology when the planet can make suggestive chemistry on its own? Now, this does not mean every Mars discovery should be poured into the Viking debate. Most rover findings have their own context and should not be forced to support an old life claim. The discipline is to use them only where they change the boundary. Organics can exist, water-shaped environments, methane remains ambiguous, and surface chemistry can be harsh. That boundary is enough. It keeps Viking connected to modern Mars without turning the story into a general rover tour. The richer Mars also changes the emotional tone of the Viking story. In 1976, the negative organics result made Mars feel chemically stripped. Today, Mars feels more like a planet with separated archives. The surface can be destructive, while deeper or older rocks preserve traces. One place can be oxidized dust, another can be ancient mudstone. One measurement can speak about the present surface, another can speak about a lake that existed billions of years ago. That means Viking's surface sample should not be forced to represent the entire planet. The landers touched what they could reach. They did not drill meters down. They did not select samples from an ancient delta after years of orbital mapping. They did not compare dozens of mineral environments. They tested accessible surface material at 2 places. That was extraordinary, but it was limited. Now limitations cut both ways. If Viking had produced no response, that would not have ruled out life in protected niches. Because it produced a response, the same limit means we cannot easily generalize it. The result belongs first to those samples under those experimental conditions. Curiosity's work in Gale Crater helps show why place matters. Ancient mudstones can preserve molecules because they formed in a different environment from the modern surface dust. A lakebed rock is not the same chemical problem as oxidized soil scraped by a 1970s scoop. When Curiosity finds organics there, it tells us Mars can preserve some carbon chemistry under the right conditions. It does not tell us what Viking's LR chamber contained. Methane, too, belongs in its lane. It's exciting because on Earth methane can be linked with biology, and because atmospheric methane should not simply last forever under Martian conditions. But possible methane changes can also come from geology or other non-biological chemistry. For Viking, methane is a reminder. Mars keeps giving us signals whose source is not obvious. That repetition across Mars science is important. We keep meeting the same shape of problem. A measurement points toward life as one possibility. Non-life mechanisms remain credible. Instruments improve. Context grows. Certainty remains hard. So Viking becomes less isolated over time, not more. It becomes the first great example of a pattern that later Mars science keeps encountering. Now, the richer Mars also protects us from cynicism. If Viking did not prove life, that does not mean the life question was foolish. Mars really did have environments that could have supported life in the past. It really does contain organic molecules in some rocks. It really does have ice, salts, and geochemical energy sources that make astrobiology worth pursuing. The search is not hype built on nothing, but it also protects us from easy belief. A planet can be habitable without being inhabited. It can preserve organics without those organics being biological. It can produce methane without microbes. It can release gas in a chamber without metabolism. The hard work is sorting those possibilities without flattening them, and that is exactly the work Viking forced onto Mars science. For a while, a simple story about Mars had power. Viking landed. The biology results were ambiguous. The chemistry instrument did not find the expected organics. Mars looked cold, dry, oxidized, and hostile. The life question seemed to retreat, but Mars did not stay that simple. Over the decades, orbiters, landers, and rovers built a richer planet. They found ancient valleys, minerals formed in water, lakebed environments, clays, sulfates, deltas, ice, salts, and rocks that recorded a wetter past. Mars became more than a dead red surface, but a planet with history. Now, history is not life. A riverbed is not a microbe. A lake deposit is not a cell. But habitability matters because it changes the background against which Viking is interpreted. If Mars had always been chemically barren in every sense, the biological reading of labeled release would look more isolated. If Mars once had long-lived water environments and preserved organic chemistry, the possibility of past or even protected niches becomes harder to wave away. The old positive-looking response does not become proof, But it no longer sits on a planet imagined as simply empty. Curiosity made that shift more concrete. In Gale Crater, Curiosity drilled ancient mudstones and found organic molecules preserved in rocks billions of years old. NASA was careful about the meaning. Organic molecules can come from life or non-life sources. Meteorites deliver organics. Geological chemistry can make them. Preservation in ancient rocks does not tell us that organisms made them, but the discovery mattered because Viking's organics problem had shaped the whole debate. If later instruments could find organics on Mars, then the old idea of a totally organic free planet looked too crude. Now be careful here. Curiosity did not prove that Viking missed a living soil. It sampled ancient rocks, not fresh Viking soil. It used different instruments. It operated in a different place and geological context. The organics it found were not a direct biological signature. They did not validate labeled release, but they did weaken the simplest dismissal. No organics anywhere, no plausible biological context. Mars also gave us methane puzzles. Curiosity reported methane variability in the atmosphere, including changing levels that have been discussed for years. Methane can be produced biologically on Earth, but it can also come from non-biological processes. NASA has repeatedly stressed that the source is unknown and that methane is not a detection of life. Again, the pattern is familiar. Interesting signal. Multiple possible sources. No proof. For the Viking story, methane is not a new main topic. We should not wander into a second documentary about atmospheric chemistry. Its role is narrower. It shows that Mars keeps producing ambiguous signals that are tempting but hard to interpret. Viking was not an exception to Mars science. It was an early version of a recurring problem. A measurement appears. The life interpretation is tempting. The chemistry interpretation is credible. The context is incomplete. The result remains bounded. And that is why modern Mars findings both help and hurt the old LR debate. They help because Mars is no longer understood as a simple sterile surface with no organic richness anywhere in its history. Ancient habitable settings existed. Organic molecules can be preserved. Water shaped the planet. Possible gas variations keep the chemistry interesting. They hurt because modern standards have become stricter. The more we learn, the less willing scientists are to let one activity signal carry a life claim. We now know Mars has many ways to make evidence ambiguous. Water can preserve records, but also alter them. Organics can be biological or non-biological. Methane can have several sources. Perchlorates can scramble heated chemistry. Surface oxidants can imitate activity, so later discoveries do not deliver Viking a retroactive victory. They deliver a more interesting courtroom. Now picture two versions of Mars. In the first version, Mars is a chemically dead desert. No organics, no meaningful water history, no preserved habitable environments, no ongoing puzzles. In that version, labeled release feels like an orphaned anomaly. In the second version, Mars is cold and harsh today, but historically wet in places, chemically complex, able to preserve some organics, able to destroy others, and still capable of producing measurements that leave scientists arguing about sources. In that version, labeled release feels less absurd, but also harder to isolate. We live with the second Mars, and that is why the pro-life case did not vanish. It adapted. It stopped leaning only on the 1976 emotional shock and began pointing to a broader Mars that might not be as barren as the early consensus implied. But adaptation is not confirmation. Curiosity's organics do not say Viking detected living metabolism. Methane variability does not say microbes are breathing under the surface. Ancient water does not mean present life responded to Viking nutrients. Every one of these findings changes plausibility, not proof, and that distinction serves the viewer better than either extreme. The old Viking found life and nobody admitted its story is too loud. The old Viking failed and Mars's boring story is too small. The real story is more durable. Mars keeps offering signals that sit between biology and chemistry, and Viking was the first mission to expose how hard that boundary is. Now we can return to the people who still think the biological reading deserves a serious place at the table. They are not arguing from emptiness. They are arguing from a signal, a control pattern, a changed Mars context, and a belief that the original life test was judged too quickly by a chemistry result later work made less simple. The question is what their strongest case still has and what it still lacks. The missing independent marker is the hinge. If Viking had found convincing organics in the same samples, the debate would be different. If a microscope had seen cell-like structures with supporting chemistry, different again. If later missions had repeated the LR pattern with modern controls and molecular support, the old argument might have transformed. But those supports did not arrive. The case survived as an argument from activity, not as a converging discovery. Here is a human reason too. Viking was the moment when the question seemed closest to a direct answer. Not an atmospheric hint, not a rock texture, not an ancient environment. A soil sample was fed inside a machine, and the machine saw a response. That is hard to let go of. But good science cannot be built on how hard something is to let go. The emotional appeal explains why people return to the result. It does not decide what caused it. That decision still belongs to evidence. So the fairest posture is respect without surrender. Respect the LR data. Respect the scientists who defend it. Respect the chemistry objections. Respect the fact that the word life needs more than a provocative curve. And that is not fence-sitting. It becomes the actual shape of the case. Here is also a philosophical strength in the pro-life argument that should be named carefully. It asks whether scientists became too dependent on the absence of organics, especially after later work showed that organics on Mars can be difficult to detect and interpret. If the GCMS result was less decisive than people once thought, then maybe the labeled release result deserves more weight. And that is not an irrational question. Scientific conclusions can shift when one pillar weakens. If a negative result was shaped by instrument limits or sample chemistry, the older balance of evidence may need adjustment. The discovery of perchlorate did exactly that for many discussions of Viking. It made the organic story more complicated, but a weakened objection is not the same as a confirmed claim. If the GCMS result becomes less absolute, the labeled release signal still needs independent support. We do not get to jump from the objection was complicated to the biology interpretation wins. The space between those statements is where the unresolved status lives. Now, proponents sometimes argue that no non-biological explanation has matched every feature of the LR data as well as biology. Critics answer that no biological explanation has supplied the missing organisms, molecules, or independent markers. Both arguments can be sincere. They are measuring adequacy differently. The biology side asks, what cause best fits the LR curve itself? The chemistry side asks, what cause best fits the entire Viking evidence set and the later understanding of Martian soil? Those are not identical questions. For a discovery claim, the wider evidence set usually wins. For a historical debate, the specific curve keeps pulling attention back, and that is why the pro-life case has survived in scientific margins and public memory. It isn't strong enough to define consensus, but it is too grounded in actual data to be treated like fantasy. It occupies a frustrating middle ground, serious, contested, incomplete. We should also remember that Viking's life tests were designed under Earth life assumptions because no one had an alternative biology to design around. If Mars life existed, would it like Viking's nutrient solution? Would water damage it? Would it be dormant? Would it live below the sampled surface? Would it use chemistry so different that a positive or negative LR result means less than either side assumes? These questions do not rescue the LR result. They widen the humility around life detection. A direct test can miss life if life does not respond. A direct test can falsely suggest life if chemistry responds. The same instrument sits between false negatives and false positives. That is a difficult place to stand, and Viking stood there first. So the pro-life case refuses to die because it has a real signal, because the original negative organic story became less tidy, and because Mars itself became more astrobiologically interesting. It fails to become proof because the signal remains one line of evidence, and because plausible non-biological mechanisms still stand beside it, and that is not a satisfying courtroom verdict. It becomes a scientific boundary. The strongest biological interpretation of Viking does not begin with aliens or drama. It begins with the labeled release data. Proponents look at the rapid response after nutrients were added. They look at the positive-looking behavior at both landing sites. They look at the way heated controls changed the response. They look at terrestrial tests where the experiment detected microbial activity in difficult soils. They look at the original criteria for what the instrument was supposed to recognize. And they say, if this had happened in an Earth soil sample, we would call it biological. And that is a powerful sentence. But it is also exactly where Mars complicates everything. Earth soil is not Mars soil. Earth controls are not automatically Martian controls. Earth microbes are not guaranteed to resemble hypothetical Martian organisms. Earth organic chemistry is not exposed to the same surface history. So the analogy matters, but it cannot be the whole case. Now, the pro-life argument has several strands. The first is kinetics, which means the pattern of the response over time. A biological reaction often has a shape, a beginning, a rise, sometimes a leveling off, sometimes a response to additional nutrient. Proponents argue that the labeled release response did not look like a random chemical burst. They see structure that resembles metabolism. The second is repeatability. Both landers saw positive-looking results. The sites were far apart. That reduces the chance that a single local oddity or instrument problem explains everything. The third is heat sensitivity. Heated controls behaved differently, and proponents interpret that as evidence that the active factor was fragile in the way life is fragile. The fourth is pre-flight testing. Before Viking flew, the labeled release experiment had been tested against Earth soils and sterilized controls. It had a working logic. It was not invented after the fact to rescue an anomaly. The fifth is the later Mars context. Ancient water, preserved organics, and a more complex planet make the biological possibility less easy to dismiss than a simple dry, sterile picture would allow. Taken together, those strands explain why the case refuses to die. But we need to be equally clear about what they do not provide. They do not provide an image of a Martian organism. They do not provide independent molecular evidence from the Viking sample. They do not show cell structures, DNA-like molecules, membranes, isotopic patterns that force biology, or a second unrelated biological marker. They do not rule out every plausible abiotic chemistry. They do not erase the GCMS problem. They do not neutralize perchlorate and oxidant explanations. And that is the missing confirmation. A serious pro-life case can be coherent and still incomplete. It can rely on real flight data and still fall short of the standard needed for a discovery claim. It can be worth discussing without becoming consensus. Here is where language matters. If we say Viking detected life, we go beyond what the evidence can support. If we say Viking found nothing, we erase the strange result that drove decades of argument. A better sentence is more disciplined. Viking's labeled release experiment produced a biologically provocative response that remains contested because chemistry offers credible alternatives and independent biological evidence was missing. That sentence is less dramatic than a headline, also closer to the truth. Now think about why wrong framings are so tempting. A clean life claim is emotionally satisfying. A clean debunk is emotionally satisfying too. Either one lets the listener close the folder, but Viking does not let us close it that way. The evidence has edges. On one edge, the signal is too structured to dismiss casually. On another, the missing organics and reactive soil chemistry are too important to ignore. On another, later Mars discoveries make the old surface story richer. On another, Modern standards demand convergence across multiple independent lines. The pro-life camp lives on the first and third edges. The mainstream caution lives on the second and fourth, and both are responding to something real. We should also be fair about scientific culture. Consensus does not form only from one data stream. It forms from how many independent routes lead to the same answer. For Viking, the routes split. Labeled release went one way, GCMS went another. Later chemistry gave the non-biological route new strength. Later habitability discoveries made the planet more interesting, but did not directly support the old sample. So the scientific community did not simply vote against an uncomfortable result. It withheld the word life because the evidence did not converge, and that is a sober outcome. But it leaves a human frustration behind. If Viking really was the only mission to ask the direct metabolism question on Mars, why not send the same test again with better controls? Why let the most provocative life detection experiment in planetary history remain unrepeated for half a century? That question is completely reasonable. Also, where the conspiracy-shaped answer often sneaks in. But the better answer is about design. Viking did not make NASA stop caring about life. It taught scientists that a direct test can be too ambiguous if the planet's chemistry is not understood first. Now we should answer the suspicion in the strongest possible way. We do not need a secret motive when the measurement problem is already hard enough. We need a test that would make the next answer clearer than the first. There is also a public communication lesson. If a second Viking-style test produced another ambiguous result, the confusion could deepen rather than clear. The world would hear life test positive again, while scientists argued over oxidants, salts, controls, and sample context. For a claim this large, repeating ambiguity is not progress enough. Progress means shrinking the number of plausible explanations. The life detection strategy also changed because Mars became a bigger scientific target. Once missions began showing ancient water environments and diverse minerals, scientists had to ask where the best evidence might be preserved. A random scoop of surface dust was no longer the only imaginable sample. Ancient lake deposits, subsurface ice, salt-rich settings, and protected rocks all became possible archives. That made mission choices harder. Do you test modern surface soil for activity? Do you search ancient rocks for preserved biosignatures? Do you map habitability first? Do you cache samples for Earth laboratories? Each path answers a different version of the life question. Viking's ambiguity pushed the field away from lonely yes or no instruments and toward combinations. That can look indirect from the outside. From the inside, it is an attempt to make the next answer harder to misread. Here is another practical issue. A repeated labeled release test would not be the same experiment unless the sample, environment, nutrients, controls, and chemistry were chosen with extraordinary care. If a new test lands on an oxidized surface and adds organic nutrients again, a positive result might only reproduce the old ambiguity. If it lands in a different place and gets a negative result, proponents could argue that Viking sites were different or that life is patchy. Either outcome might leave the central argument alive. So a repeat has to be more than a replay. It would need to ask why the old result happened. That means measuring the oxidants before feeding the sample. It means detecting organics without destroying them. It means understanding perchlorate and related salts. It means running controls that separate chemical reactivity from biological metabolism. It means choosing samples where life, if present, has a plausible chance of survival. Those requirements turn a simple question into a large payload. Now add mission constraints. Every gram on a lander matters. Every instrument competes with other instruments. A Mars mission is planned around risk, power, mass, thermal limits, communication, landing safety, and years of engineering. Scientists do not get to send every experiment they want. They have to choose the set most likely to produce interpretable results. After Viking, interpretable became the key word. A dramatic answer that cannot be interpreted is not enough. It may be historically fascinating, but it does not close the science. So that is why Mars exploration leaned toward context: geology, mineralogy, atmospheric chemistry, organic detection, and eventually sample caching. Now, could that strategy have been too cautious? Possibly. Reasonable people can argue that direct life detection should have returned sooner with improved designs. The absence of a clean modern repeat is part of why Viking remains emotionally open. A new mission built around the old ambiguity would be valuable. But caution is not the same as avoidance. It can be the result of learning. The best analogy is medical testing. If one early test gives an alarming but ambiguous result, the answer is not always to repeat the same test forever. Doctors order different tests. They look for independent markers. They check imaging, blood chemistry, symptoms, history, contamination, and false positives. The goal is not a louder signal. The goal is a diagnosis that cannot be easily mistaken. Mars life detection moved in that direction. So when later missions studied ancient habitable environments, they were not changing the subject, they were building the missing context. When they searched for organics with better instruments, they were addressing the Viking contradiction. When sample return became a goal, it reflected the need for Earth lab confirmation beyond what a small landed instrument can do alone. The public question remains impatient, and understandably so. Did the old test see life or not? Science answers more slowly. First, we have to know what else can make the test look that way. That slower answer is frustrating. Also, why the next direct test, if it comes, may finally be worth believing. The simplest question after Viking is also the most annoying one. Why not do it again? If the labeled release result was so provocative, if the debate mattered so much, if the direct life test remains unmatched in public imagination, why did later Mars missions not simply fly a better version, scoop up soil, add nutrients, and settle the issue? And it is a fair question. But the answer is not secrecy, it's measurement design. Viking revealed that a metabolism-style test can produce a result that looks exciting and still be scientifically unsafe. If unknown soil chemistry can mimic biology, then repeating the same basic idea without first understanding the chemistry might create a second, ambiguous answer. Better instruments could help, but the deeper problem would remain. One activity response is not enough. Now look at what later Mars missions actually did. They chased water, they mapped minerals, they studied ancient environments, they searched for organic molecules with improved instruments, they explored whether Mars was habitable in the past. They examined rocks that could preserve biosignatures. They built a context around samples before asking what those samples might mean. That strategy can look indirect if the viewer wants a yes or no life test. But it makes sense after Viking. If you do not know whether the soil itself can counterfeit metabolism, then you need to understand the soil. If surface oxidants can destroy organics, you need to know where organics might survive. If heated chemistry can alter the molecules you want to detect, you need better analytical methods. If one instrument can be fooled, you need multiple independent signatures. So later missions were not a retreat from the life question. They were a move toward evidence that could survive the Viking problem. Now consider the difference between habitability and life detection. Habitability asks whether an environment could support life. Was there water? Was there energy? Were the right elements present? Did conditions persist long enough? Life detection asks whether life is or was actually there. The first question is easier to support with geology. The second requires much stronger evidence. Mars exploration after Viking spent decades strengthening the first question because Viking showed how dangerous it was to leap into the second. Now, that does not mean everyone agreed with the strategy. Some scientists and advocates argued that direct life detection should have returned sooner. They believed Viking's result deserved a cleaner repeat. They worried that habitability studies could become a long detour around the most interesting question. That criticism has emotional force. A viewer can feel it. We sent a life test. It gave a provocative answer. Then we spent decades talking about ancient water and minerals. Why not ask again? But the mission planning answer is heavy. Spacecraft are expensive. Payload mass is limited. Planetary protection is demanding. Instruments must be chosen years before launch. A direct biology experiment that produces another ambiguous result may fail to justify the cost if it cannot overcome the core uncertainty. So the standard changed. A modern Mars life claim would need evidence that converges. A metabolism-style response would need molecular chemistry beside it. Organic detections would need geological context. Possible biosignatures would need contamination controls. Samples would need to be understood in place, not as anonymous powder in a cup. Ideally, some of the most difficult analyses would happen in Earth laboratories, where instruments can be larger, more flexible, and more carefully cross-checked. And that is why perseverance matters in this story, even though it is not a Viking repeat. Perseverance is collecting and caching samples from an ancient river delta environment in Jezero crater, chosen because it may preserve records of past habitability and possible biosignatures. The sample return vision is not a direct metabolism test on the surface. It becomes a convergence strategy. Bring carefully selected material to the best laboratories on Earth and ask many questions at once. As of this script, the sample return effort has faced delays, redesigns, and budget challenges. But the scientific logic remains important. The answer to Viking's ambiguity is not make the old test again. It is build a result that chemistry, contamination, and context cannot easily explain away. Now we should not pretend this fully satisfies everyone. A protected subsurface sample with modern metabolism assays could be valuable. A new lander designed explicitly around oxidants, perchlorates, organics, and multiple controls might finally answer whether any present-day Martian material behaves biologically. Many astrobiologists would love sharper life detection tools on Mars. But the reason they have to be sharper is Viking. Viking showed the danger of asking a living question of an unknown chemistry. It did not show that direct tests are useless. It showed that direct tests must be paired with enough context to prevent a false positive from becoming a scientific trap. And that is the better answer to the suspicion. NASA did not stop because the answer was hidden. The strategy changed because the first answer could not be interpreted safely. And if a modern mission ever asks Mars a direct life question again, it will have to be designed around every way Viking might have been fooled. If we imagine sending the perfect life test tomorrow, the first requirement is not drama, it is patience. We would need repeated measurements across samples, seasons, depths, and chemical settings. We would need to accept that Mars may not give the same answer everywhere. A result in oxidized surface dust may differ from a result in protected ice-rich material. A delta rock may preserve an ancient trace while a modern soil assay remains silent. That variety is not an inconvenience. For us, it may be the only way to understand the planet, and the final standard may be social as much as technical. A life detection has to persuade people who want it to be true and people who expect false positives. It has to work across laboratories, teams, instruments, and interpretations. It has to survive the excitement of the first announcement and the skepticism of the 10th reanalysis. Viking did not fail at social standard because the scientists were weak. It failed because the evidence was not built to carry that much weight. A modern test would also need to be honest about negative results. If no activity appears in one surface sample, that cannot mean Mars has no life anywhere. If no organics appear in a harsh oxidized sample, that cannot mean organics never existed. If methane is absent during one measurement window, that cannot close the atmospheric question forever. So the experiment must be designed around bounded conclusions. It should be able to say, under these conditions, in this material, with these controls, this response did or did not occur. That sounds modest, but modest claims are the building blocks of durable discoveries. Viking's mistake was not that it asked too modest a question. In some ways, it asked too large a question through too narrow a channel. A modern mission should keep the directness while widening the evidence around it. A modern mission would also need to think carefully about water. Viking added a liquid nutrient which made sense from an Earth biology perspective. But Mars' surface life, if it exists, might be adapted to extreme dryness or salts. A sudden wetting event could stimulate organisms, damage them, or trigger non-biological chemistry. The same action could help life reveal itself or destroy the very pattern we hoped to see. So modern tests might vary water activity instead of simply adding liquid. They might test tiny humidity changes. They might compare brines, dry nutrients, vapor exposure, and carefully controlled temperatures. They would ask not only whether the sample responds, but under what physical conditions the response appears. Temperature would need the same care. Heating can sterilize, but it can also transform chemistry. Cooling can preserve, but it can slow metabolism. A modern experiment would not treat temperature as a blunt switch between alive and dead. It would use temperature as a diagnostic tool, watching how different reactions change across a range. Now add time. Viking watched on mission timescales. Future instruments might need to monitor slower responses, dormant organisms, or repeated cycles. Some life processes might not respond instantly to unfamiliar food. Some chemical reactions might fade after an initial burst. The shape over hours, days, and repeated treatments could help distinguish causes. The sample preparation would matter too. Crushing, scooping, exposing, sieving, heating, and wetting all change material. A modern test would need to know whether it is testing the original environment or a disturbed version created by the instrument. For a fragile biosignature, disturbance can be fatal. For reactive chemistry, disturbance can create misleading activity. Here is why in-place context and returned samples complement each other. A lander can test fresh material under Martian conditions. Earth labs can analyze tiny structures and molecules with instruments too large and complex to fly. Neither path alone is perfect. Together they can cross-check each other. A modern life detection program might also need humility about alien biology. If life on Mars shares ancestry with Earth life through meteorite exchange, it might use familiar molecules. If it began independently, it might not. An experiment that assumes too much Earth-like behavior could miss a stranger biosphere, but an experiment that assumes anything at all can become impossible to interpret. So the goal is not to design a detector for every imaginable life. The goal is to build a result where several independent signs line up in a way non-life struggles to mimic. Activity, molecules, patterns, environment, controls, replication, contamination exclusion, independent confirmation, those are not bureaucratic hurdles. They are how a claim survives. And there is one more requirement. The result must be able to disappoint us. A good experiment is not built only to produce the answer we want. It is built so that a negative result means something, a positive result means something, and an ambiguous result teaches us what to fix. Viking's ambiguous result was painful because it sat between those categories. A modern test should narrow the space where ambiguity can hide, and that is a high standard. But after Viking, it is the right standard. A modern life test on Mars cannot be a single instrument waiting for a single dramatic signal. It would need a ladder of evidence. The first rung could still be metabolism. There is nothing wrong with asking whether a sample transforms nutrients, releases gas, consumes molecules, or changes chemistry in ways that suggest activity. Living systems do things. A direct activity test can still matter. But one metabolism-style response would not be enough. The test would need multiple independent assays, different nutrients, different chemical pathways, different controls, different temperatures, different water activities, different sample treatments. If biology is the cause, the pattern should have a coherence that random reactive chemistry struggles to imitate. Now add the second rung, organic chemistry. A modern mission would need to know what molecules are in the sample before, during, and after the activity test. Are there organics? What kinds? Are they simple compounds that could come from meteorites or geology, or more complex patterns that suggest biological selection? Are there repeating molecular families? Are there compounds that survive in protected niches? Do the molecules change in a way that matches the activity signal? Here is where technical ideas like chirality can matter, but we should translate them. Many biological molecules come in left-handed and right-handed forms, like gloves. Life on Earth strongly prefers certain handed forms. A strong imbalance in the handedness of relevant molecules could be interesting, though not automatically decisive. A modern test would treat that as one clue among several, not a magic stamp. The third rung is chemistry control. Any modern Viking-like experiment would have to account for oxidants, perchlorates, and other reactive salts. It would need control runs designed not only to kill life, but to isolate non-biological reactivity. Heat controls alone would not be enough. The mission would need chemical blanks, sterilized samples, altered humidity, treatments that neutralize oxidants, and ways to test whether the same gas release occurs without a plausible biological pathway. In plain language, the instrument would have to ask, is this sample eating, or is it just chemically attacking the food? The fourth rung is sample context. Viking took surface soil. That was understandable. It was the material the lander could reach. But the Martian surface is harsh. Ultraviolet light, dryness, radiation, and oxidants make it one of the hardest places to preserve delicate evidence. A modern test might prefer protected material, shallow subsurface samples, ice-associated material, recently exposed deposits, ancient mudstones, salt environments, places where organics are more likely to survive and where harsh surface chemistry is reduced or at least understood. The sample cannot be anonymous dust. Its story matters. Where did it come from? What minerals surround it? Was it once wet? Is it exposed to radiation? Does it contain salts? How old is it? Was it altered by water? Could organics survive there? Is it a place where life would plausibly live now, or only a place where past life might have left a record? The fifth rung is contamination discipline. Here is less glamorous than a signal, but it is essential. A life claim can be ruined by Earth contamination. Spacecraft carry molecules, materials, and possible biological traces unless cleaned and controlled with extreme care. If a mission feeds a sample and sees activity, scientists must know that the activity did not come from something the spacecraft brought. Planetary protection works in both directions. We do not want to contaminate Mars with Earth life. We also do not want to bring a sample back, if that happens, without proper containment and care. Any serious life detection program has to treat contamination as part of the science, more than a procedural burden. The sixth rung is independent confirmation. A signal should not stand alone. If a metabolism assay is positive, molecular chemistry should support it. If organics look interesting, isotopes might help. If isotopes look interesting, microscopy or mineral context might help. If a possible biosignature appears in one sample, related samples should be checked. If an instrument makes a claim on Mars, Earth laboratories should test returned material where possible. No single rung carries the word life by itself. That may feel frustrating. The human mind wants a clean sign, a living cell under a microscope, a gas spike that means metabolism, a molecule that means biology, a yes or no light on the dashboard. But life detection on Mars is more like building a net. Each line catches a different way the answer could escape. Now compare that to Viking. Viking had brave instruments and real controls, but it lacked the modern context. It did not know about perchlorate in Martian soil. It did not have rover-based geological histories. It did not have Curiosity's organics record. It did not have modern sample handling expectations. It did not have the ability to drill deeply into carefully selected environments, cache samples, or run Earth Lab follow-up. Viking was not foolish. The mission was early. That may be the fairest description of the mission. Early enough to ask the direct question. Early enough to be surprised by the chemistry. Early enough to make a result that still mattered, but not equipped to close the interpretation under modern standards. So if a future mission runs a new direct life test, it should not simply imitate Viking with better electronics. It should ask a more armored question. Does the sample show activity? Do the molecules support that activity? Do the controls rule out oxidants and perchlorates? Does the environment make the result plausible? Is contamination excluded? Do independent methods agree? Can the results survive skeptical laboratories on Earth? Only then does a signal begin to approach a discovery. And even then, scientists would be careful. That caution is not cowardice. It is respect for the size of the claim. Life on another planet would be one of the most important findings in human history. The word cannot be carried by excitement alone. It has to be carried by evidence strong enough to outlast the first wave of disbelief. And for us, that changes the meaning of patience. We are not waiting because the question is weak. We are waiting because the standard has to be strong enough that the answer still stands after the thrill is gone. Viking gave us the first great warning. A planet can answer in a way that sounds alive before we understand the language it is speaking. Now, with that standard in mind, we can finally return to the title and answer it honestly. Maybe the most useful phrase is not life detection, but life interpretation. Detection sounds like the instrument simply sees the answer. Interpretation reminds us that the instrument sees evidence, and humans decide what the evidence can support. Viking detected a response. It did not secure the interpretation. The unresolved status also protects future science from two temptations. One temptation is dismissal. Viking was old. The instruments were primitive. Forget it. The other is inflation. Viking already solved the life question, and caution is denial. Both temptations flatten the evidence. Viking was old, but not trivial. Its instruments were early, but not careless. Its result was provocative, but not decisive. Its ambiguity is not a failure of imagination. It becomes a real measurement problem created by a real planet. And that is why the best final answer has to feel balanced without becoming bland. Viking gave us a signal worth respecting and a warning worth obeying. The word unresolved can sound weak, as if science has failed to make up its mind. But in this case, unresolved is a disciplined status. It means the result has not been dismissed as meaningless, and it has not been elevated beyond its support. It remains alive as a question because both sides have evidence worth weighing. And that matters for how we picture Mars itself. We are not looking at a neat moral tale about brave scientists or timid institutions. We are looking at a world whose surface can produce just enough signal to force belief and just enough chemistry to block it, and that is different from mystery for mystery's sake. Some mysteries survive because people enjoy them. Viking survives because the measurement record and the interpretation problem are genuinely difficult. Now, what can we say with confidence? We can say Viking operated direct biology experiments on Mars. We can say the labeled release experiment produced positive-looking responses. We can say the GCMS did not provide the expected organic support. We can say Martian surface chemistry, especially oxidants and later perchlorate-related understanding, makes non-biological explanations credible. We can say later Mars missions found a more complex planet than the simplest early story allowed. We can say no independent biological confirmation has closed the case. Those statements form the boundary. Inside that boundary, different scientists can lean differently. Some see the LR data as biology that was never given its due. Others see a classic false positive from reactive soil. Many see a historically important ambiguity that shaped astrobiology more than it answered it. For the viewer, the value is not choosing a team. The value is understanding why the answer is hard. Viking asked Mars a question with the tools and assumptions of its time. Mars answered in a way that sounded partly biological and partly chemical. The rest of the field has spent decades learning how to ask better, and that is why the title has a careful answer. Yes, NASA already ran the direct life test that still defines the debate. No, that test did not give us life on Mars as a settled discovery. The most honest space between those answers is where Viking still lives, and maybe that is why the story remains so compelling. It isn't about a failure to find wonder, it becomes about the difficulty of proving wonder when chemistry is clever enough to imitate it. Viking, did NASA already run the only real life test on Mars? In the narrow sense that matters for this story, yes. Viking ran direct biology experiments on Martian soil. It did not only study whether Mars had once been habitable, it did not only map minerals or search for ancient watery environments. It placed samples into laboratory chambers and asked whether they would behave like living chemistry. And that is why Viking remains unique. But did Viking prove life on Mars? No. The labeled release experiment produced a real and provocative response. It was positive looking. It appeared at both landing sites. It had timing and control behavior that serious proponents still argue can fit biology. It deserves to be treated as one of the most consequential unresolved measurements in astrobiology, but proof requires more than that. The GCMS did not find the expected organic support in the Viking surface samples. Reactive oxidants made false positive chemistry plausible. Perchlorate discoveries later gave scientists a stronger physical mechanism for how Martian soil could alter organics and complicate heated analyses. Modern Mars findings made the planet more interesting, but they did not independently confirm that Viking had detected metabolism. So Viking sits in an unusual place, and it is not a dead failure, it isn't a confirmed discovery. It becomes an unresolved experiment that exposed the boundary between a signal and an interpretation. Now let's say the core lesson plainly. A life detection instrument can work and still leave us uncertain. That sounds paradoxical, but Viking shows how it happens. The instrument can measure the thing it was built to measure. Gas can be released, controls can differ, data can be valid, and still the cause can remain disputed because another explanation fits enough of the same facts, and that is the heart of the story. If biology caused the labeled release response, then Viking saw one of the most important signals ever measured by a spacecraft. If reactive soil chemistry caused it, then Viking discovered something almost as important for future life detection. Mars can imitate biology well enough to fool a direct test. Either way, Viking mattered, and that is why the result keeps returning. It lives in the gap between two honest statements. The signal should not be dismissed. The signal should not be crowned. Now consider what Viking changed. Before the mission, it was possible to imagine that a lander could carry a life test, run it, and give humanity a clean answer. After Viking, that innocence was gone. Scientists had to ask harder questions. What counts as metabolism on another planet? How do we test unknown life without assuming Earth biology? How do we separate living chemistry from oxidized minerals? How do we interpret a sample when the surface itself can attack the compounds we add? How many independent lines are enough? Those questions shaped Mars exploration for decades. They also changed the emotional meaning of the search. Mars life became less like finding a creature and more like proving a pattern. Not one sign, but a web of signs. Not one instrument, but convergence. Not one dramatic moment, but a chain of evidence strong enough to survive the chemistry of a different world. That may feel less satisfying than the old dream, but it is also more mature. A future discovery, if it comes, will owe something to Viking. It will be more careful because Viking was ambiguous. It will control for oxidants because Viking exposed them as a problem. It will treat organics with more sophistication because Viking's GCMS result became so contested. It will demand multiple lines because labeled release showed how powerful and dangerous a single response can be. So the Viking result is more than an old argument. It becomes a design lesson still sitting on Mars, and it leaves us with a bounded verdict that is stronger than either myth. NASA did run the clearest direct metabolism-style life detection experiments ever successfully operated on Mars. One of them produced a response that still deserves serious attention, but Viking did not deliver the independent evidence needed to call that response life. The question remains unresolved, not because anyone needed it hidden, and not because the data were meaningless. It remains unresolved because Mars gave a real signal inside a chemical environment we did not yet understand, and that is the experiment's legacy. Viking may not have found life, but it may have found the reason life detection is so hard. Now we can hold Viking in a clearer form. It was a physical experiment on Martian soil, and Mars answered in a way the mission could not fully decode. That answer matters because it was not empty. The labeled release experiment produced a metabolism-style response at 2 places on Mars. It changed when the sample was heated. It was strong enough that serious scientists still argue over it. But the limit matters just as much. The supporting organic evidence was missing by the standards of the time. The soil later proved more chemically strange than Viking's designers could fully know. Perchlorates, oxidants, ultraviolet damage, and reactive minerals all give Mars more ways to imitate activity without requiring a cell. So the honest verdict is narrower than the legend and stronger than dismissal. Viking showed that a direct life test on Mars could produce a real, provocative signal. It did not show that the signal had to be alive, and that is the lesson every future mission inherits. A life detection experiment can not only ask whether a sample reacts, it has to ask what else can make the same reaction, what molecules support it, what controls survive it, and whether the result holds when the first excitement has passed. Viking did not give us a dead end, it gave us the shape of the problem. A planet can answer loudly and still not answer clearly. That is hard to sit with, because we want the universe to use labels we can read. We want life to announce itself with one unmistakable signature. Mars did something less convenient. It gave us an experiment that still forces us to separate reaction from explanation and signal from proof. If you imagine standing beside that little chamber in 1976, the lesson is immediate. The machine was not useless because the answer was ambiguous. The ambiguity was the discovery. The cleanest discoveries are usually remembered as moments. A telescope sees a world. A detector catches a particle. A rover drills a rock. But behind each moment is a standard of evidence, and Viking changed that standard for life beyond Earth. After Viking, a direct response was no longer enough by itself. The response had to be surrounded by chemistry that made sense, controls that survived skepticism, and context that narrowed the ordinary explanations. And that is not a smaller dream, it becomes a stronger one. A life claim that survives those demands would mean more, not less, because it would not depend on our excitement to stay alive. It would stand even after the chemistry had been tested, the samples had been compared, and the alternative explanations had done their best. And that may be the most valuable legacy a first experiment can leave. Viking's answer was not final, but it made every later answer more honest. And that is a quieter ending than a discovery headline. Also more durable, because the next real claim will need exactly that discipline to stand. The old lander did not give the answer people wanted. It gave the discipline the answer required. Be curious, be skeptical, and never confuse a signal with the full story of its source. And that is why the title keeps its force. The answer is more than historical. It changes how we hear every new possible biosignature. When a mission finds carbon, we ask about source. When it finds gas, we ask about chemistry. When it finds an environment that could support life, we ask whether anything actually lived there. Viking trained us to keep those questions separate, even when we wish they would collapse into one. So when the old lander appears in this story, it should not be treated as a relic. It's still one of the sharpest mirrors we have for the search for life beyond Earth. It shows our impatience, our hope, our caution, and our dependence on instruments that must translate alien chemistry into human confidence. The next great astrobiology claim may come from Mars, Europa, Enceladus, Titan, an exoplanet atmosphere, or a sample returned from somewhere we have barely imagined. Wherever it comes from, Viking's lesson will be waiting underneath it. A signal is the beginning. The hard part is earning the interpretation. A quiet humility sits in that ending. The first direct question was not wrong, it was early. It reached for biology before Mars had taught us enough about its soil. Now every future claim has to carry that memory. A gas release is not enough. A molecule is not enough. A tempting pattern is not enough. Even a beautiful signal has to be placed beside the ways a planet can deceive our expectations without any intention at all. For us, that means patience has become part of the evidence standard. We have to let the chemistry argue back before we let the conclusion harden. And if one day Mars gives us a stronger answer, Viking will still be part of it. The new result will be designed in the shadow of the old one. It will know to distrust simple activity. It will know to measure the chemistry around the signal. It will know to ask what heat, water, salts, oxidants, and contamination can do. It will know that the most exciting interpretation is not always the only one. That does not make the search colder, it makes it more serious. The next natural question is what Mars looked like when liquid water and usable chemistry had more time to work.